So I hope everyone had a good break. You remember what we were talking about before break? We talked about toxicity, environmental toxicity. We defined lethal dose and lethal concentration. And I mentioned sublethal toxic effects. And so today we're going to focus on those and especially how we might quantify sublethal toxic effects in aquatic ecosystems using the ecosystem services concept. And then we'll talk about some specific heavy metals that are listed here. Hopefully I'll get through all of them. And as last time, if I go a little bit over, I'm not sure I will, but if I do, for the purpose of the video, I'll just keep talking. If you have to leave, that's okay. But I also want to remind you that you should be finishing up the reading because we have one more topic in the sort of hydrologic cycle component of this class, which is the next two lectures after this, which have to do with water purification and sewage treatment. And then we're moving on to the atmosphere. And your midterm will cover up through the sort of heavy metal contaminants portion. So you really need to finish up the reading if you haven't already. Okay, so the first thing I wanted to talk to you about is how we might use ecosystem services in a broad sense to quantify sublethal toxic effects. So rather than die-offs of organisms because of exposure to high concentrations of some contaminant, we're talking about things that impact the way an ecosystem functions. And I'm going to highlight one meta-analysis that looked at a whole bunch of different studies that put together kind of different contaminants and different foundation species. And foundation species are things like coral in a coral reef, or kelp in a kelp forest, or trees in a terrestrial forest, things that have to be there for that habitat to even exist. And the important thing is that whether or not lethal or sublethal concentrations, the impacts can affect habitats, right? And they can diminish the services that the habitats provide. And they're especially problematic when they affect the foundational members of an ecosystem. So this is just a reminder about ecosystem services. I think I've mentioned them once before during the class, but there's lots of different ways that we can think about what ecosystems provide to us. This is a sort of a quantifiable economic way of doing it. It may not appeal to your sense of the aesthetics or the aspects of an ecosystem that are unquantifiable, but it is a way of quantifying what things an ecosystem provides and that can be impacted by human activities. So things like regulating services, carbon dioxide coming out of a forest, cultural services, people using an ecosystem for part of their traditional culture, whether it's indigenous or otherwise. You could even think about if you like to go hiking in the hills, that's a cultural service. Supporting services, things like rainforest here, providing the ability for water to fall on the land surface to replenish our aquifers, which provides us with drinking water. And then there's what are called provisioning services, which are oftentimes the ones that people focus on because these are economic, taking of organisms from the ecosystem and selling them for something. So all of these are different quantifiable measures. And there are other ones too. There are aesthetic services, for instance. You could add to this list, but these are the ones that are sort of quantifiable and then can be compared from one to another, both in terms of the health ecosystem and the impacts. And so, I would challenge you to think about, pick your favorite ecosystem. It could be the Amazon rainforest, it could be a desert, it could be anything. Just think about these issues. And oftentimes, irrespective of human impact from pollution and stuff, it's this which takes over for some of these others. When a ecosystem is overutilized for its provisioning services, then it can have negative impacts on some of the other things. So we want to think about how sublethal toxic effects affect the ecosystem services. And as this graphic kind of shows, it's a little bit complicated. When you go into an ecosystem that is unperturbed and you're sure that it's unperturbed, you can sort of try to define the services. But that doesn't necessarily mean that that is the state that it always is. Think about an ecosystem that requires a lot of humidity and warmth that is at the edge of the sort of tropics during an ice age. It may change for natural reasons as we go into a cool and a warm period. So we need to think about the state of the ecosystem. And then there are the human impacts, such as how have humans transformed an ecosystem? What are the economic benefits of the transformed or untransformed ecosystem? And what's the value? And this conceptual framework, which is basically thinking primarily about these provisioning services, is, I mean, like, to me, I kind of don't like it in the sense that I think a healthy ecosystem provides many other benefits other than the economic ones. But this is a way that ecologists can demonstrate with a fairly impartial kind of way what the negative potential impacts are. And some of these things are relatively complicated, especially if the ecosystems have been transformed. And so you can quantify them. You can quantify them in monetary terms. You can also quantify these kinds of things in physical terms, like loss of habitat, for instance. But it's to be able to compare. This is oftentimes, or to look at how something has evolved in time because of human impacts. This is commonly how it's done. And if you want to read more about it, you can read about it. That's for a coral ecosystem in that paper. So when we think about ecosystems, I think there's some kind of integrity that is has some bounds on it, right? That not all coral reef ecosystems are exactly the same, even if they're not impacted by human activities. But there's kind of a range of conditions, and then we can think about that range of conditions in sort of physical ways, biological ways, and chemical ways. And then if we further are able to show that human impacts are affecting one or the other of these things, or all of them, then we can quantify that and be able to demonstrate, for instance, sublethal toxic effects are having an impact on an ecosystem. So this is the results of a meta-analysis. Everyone knows what a meta-analysis is, where someone takes a whole bunch of different studies and puts them together and pulls more out of it than the individual studies. And this is from 3 years ago, and I've given you the reference here if you wanted. There's this, and it continues on to the next slide. It's a rather complicated slide. So let me just walk you through it. There are different parameters that are looked at in different ecosystems where they're looking at the foundation species of the ecosystem. So corals in a coral reef, bivalves in a clam bed, for instance, kelp in a kelp forest, fucoids are a kind of brown seaweed, mangroves in a mangrove swamp, salt marsh plants, sea grants plants, etc. So that's the symbol. So for instance, if you saw a circle here, well, that's a coral. It's color coded by what the contaminant type is. And again, this is from a whole bunch of different studies that were included together. So we've got polycyclic aromatic hydrocarbons, organic compounds, metals, herbicides, multiple metals together, and then other, meaning things that are not easily fit into any of those categories. And so then on this chart, you'll see sort of a dot, which is the average of all the studies, and then this bar is sort of the range. 0 means there's no negative or positive impact. And if you find things that are over here, and of course, the bigger the number, the bigger the impact, that's a positive impact. The things on this side are negative impact. And so what these authors have done is gone for little asterisks by the ones that are significant, meaning they don't overlap 0 in any case. For all of the various studies that are wrapped up into this particular symbol, for instance, there's always a negative effect. Sometimes it's really negative, sometimes it's not so negative, but it's always negative. So you can see that, for instance, some of these things, like in pink, those are having multiple metals together in fucoids, that sometimes has a positive effect. These are some of the synergistic effects we talked about last time, like when we talked about adding lead nitrate, and it was the nitrate was allowing a seagrass to grow, more so than the lead was impacting the growth. And so from this, you can see this is the efficiency of photosynthesis. So this would only be for the organisms here that do photosynthesis. That's why you won't find bivalves in here. This is leaf area. That's obviously for only the organisms that are leafy. And then we've got respiration for the respiring, filtration, growth, and chlorophyll A. And so you'll see here that, for instance, again, the multiple metal seems to have a positive synergistic effect. Growth for almost all of these different things that are listed here, and almost all these contaminants, this brownish color is herbicide effect on seagrass, for instance, we see negative effect. But not everything shows a negative effect. So this gives us the ability to start to focus in on which contaminants and which environments should be focused on, meaning it doesn't mean that you couldn't have a negative effect on one of these other scenarios, but these are more likely. So these are the things that people tend to look for. So this is some plots of the size of the effect in some sort of quantifiable way versus the concentration of the contaminant. And again, same symbols over here, but now this is just focusing on heavy metals, which is sort of the topic of what we're talking about today. So each color is a different heavy metal. And what you'll see is, and I just want to highlight where zero effect means, baseline. Basically, there is no measurable effect. And so you can see that for some of these things, there's a really strong effect that continues to higher and higher concentrations. For other of these, there's basically an effect at a low concentration that then just continues to be present, except for in one study where there was a much lower effect. If you also recall, last time we looked at biomagnification and we looked at the concentration of mercury in food fish and bird feathers, and we saw that it saturated out at some point. So that if there is a strong sublethal toxic effect that reaches toxicity, you can get populations like this that then just they don't continue to higher concentrations because they don't exist in the higher concentrations. And so, like I say, in this particular plot, photosynthetic efficiency doesn't seem to be affected that much. Growth doesn't seem to be affected that much, but all these other parameters show a pretty strong effect for a variety of different metals. And then this is just some specific, this is for silver, copper, and cobalt for some habitat formers. And this is copper and zinc, specifically in fucoids and salt marsh plants. And you can see that in all these cases, there is a pretty measurable negative effect. This is the only panel that is repeated from the previous slide, but they take different forms. And so this means that while we can make some generalizations, if you really want to study this in a particular ecosystem, you have to get into more detail. This is a summary of that. So basically, this set of columns for this and this are for corals. These are for bivalves. These are for, I believe that is kelp, but it could be a fucoid. And the point of this diagram is to show you the parameters, right, up here, chlorophyll, photosynthetic efficiency, growth rate, etc. And down here, the various contaminants, herbicides, polycyclic aromatic hydrocarbons, metals, etc. And these are repeated. And we will talk about TBT in a second, tributyl tin, a particularly gnarly contaminant that used to be painted on boat hulls to prevent things from growing on the boats. And so if you look at the scale, the lightest color means there's a negative impact. That medium gray means it's indistinguishable from baseline, and brown or dark gray means there's a positive impact. So I think you can see that for most of these cases, there is, for some of these parameters, for instance, there are cases where there doesn't really seem to be much impact. That aspect for those foundational species, corals and a coral reef, they don't really have much effect on productivity or respiration, for instance. Chlorophyll A doesn't seem to be that much affected. Interestingly enough, though, the photosynthetic efficiency is. And so, and if we move over to bivalves, their white bars are bigger in proportion. And then back over here into the kelp, it's a little bit more complicated. But when we look at the individual contaminants, we can see several of these bars are almost entirely white, right? That means that for those things, they can potentially have a very strong negative impact on those particular organisms. And for instance, as I say, we'll talk about tributyl tin today. There's in no case, no study where it didn't have a negative impact, meaning there aren't even any of these sort of neutral cases. So this is, like I say, one way of kind of assessing. This is just a focus in on the corals, just because I thought they were relevant to us here. This is a repeat of what was before. I think what on the last slide, what you can see is that herbicides are potentially very, very significant in their impact. Polycyclic aromatic hydrocarbons and metals, not as much. Organic compounds, depending on what they are and where they are, and in which cases are more likely to have a negative impact than not, because this bar is less than 0.5 and there's no positive impact. Others is sort of generic, other types of contaminants that are not specified in the paper also can have a pretty significant impact. And the two things that are impacted the most are the efficiency of photosynthesis and the number of zooanthellae, which zooanthellae are the symbionts that live in the coral that allow it to do photosynthesis. Okay, so this is kind of in a nutshell, a brief description of how we can quantify sublethal toxic effects by using this sort of ecosystem services concept. So now, the rest of the time, we're going to talk about some specific metals. We've already talked a little bit about mercury, so I'm going to go through it pretty quickly, but then we'll talk about these other metals too, and some of the particular considerations. So for mercury, I want to talk about catastrophic contamination as opposed to sort of the baseline background contamination, which we've already talked about. I just want to remind you of how mercury gets into food webs, right? So we've talked about mercury being imposed into the environment, being transformed, especially by sediment-dwelling microbes, into fat-soluble forms, and then getting up into the water column and being accumulated by either filter feeders living at the sediment-water interface or plankton in the water column, and then getting into the food web and being passed up. And so these organisms at the base of the food web are responsible for accumulating a lot of the mercury that's in the environment. And we've talked about this. I'll remind you on another slide a little bit about the water and the sediments and everything else. But once it gets from this, it can then move up into food webs, ultimately reaching humans and other mammals where the concentrations can be quite high because of the concept of biomagnification. So if you recall last time we talked a little bit about this methylation that happens in sediments that makes this mercury more volatile as well as fat soluble, which allows it to be accumulated. And in a lot of background settings or baseline settings where there's a lot of petroleum burning, coal burning, other industries that use a fair amount of mercury, there's kind of smokestack emissions that accumulate over time and get up into food webs. So we're going to talk about more of an acute contamination where there was a large mercury spill. And interestingly enough, this same sort of ecosystem model applies. The same bioaccumulation and biomagnification happens. So this is infamous, I guess you would call it, Minamata Bay. So toxic acute mercury poisoning is called Minamata Bay disease because of this case that happened in post-World War II Japan, where especially back in those days, the diet was very much based on seafood and things taken from the ocean. And it turns out there was mercury sulfate that was being used as a catalyst in a manufacturing plant, and they dumped a ton of it into a local river, and it went from that local river into the ocean. And so all of these, triangles and dots are cases that were, fatal. And so, we have ones in babies and we have children and adults, and they're all sort of focused around one area. There's a couple of other outlying areas. And then sometime later, this was, I wrote down when this was. This was in the 50s. And then sometime later in the 60s, there was another occurrence elsewhere in Japan, not as extreme, but still. And it took a little bit of time to figure out exactly what had happened and why. but it was the introduction of really high concentration of mercury, a very rapid accumulation into the food web, and sort of reliance on marine resources as part of the food for that group of people that they got so sick. And I mean, interestingly enough, mercury is still a problem, and especially even in Japan. So there's, because of legacies, it's not that they're currently using mercury in a way or in such high concentrations or contaminating the environment, but once you put it there, it stays for quite some time. And so, for instance, there was this kind of well-known case, recently, within the last decade or so, where mercury was found to be very high in food fish, even food fish being served to school kids. And there was one particular, fish that was taken from this bay. This bay is famous, it's Taiji. It's a place where every year, as part of a cultural practice, they sort of like all the fishermen of the village, like, get a whole bunch of organisms together into this bay, and then they kind of spear them all and kill them. And so, one of these fish, which had been used for food fish for schools, had something like 11 ppm of mercury, which is just way, way, way too high. But it also had polychlorinated biphenyls in it, which is another thing that can bioaccumulate in marine food webs. And so, this is significant because of the numbers of dolphins that are consumed. And so after this, the Japanese put in some kind of controls where they measure the amount of mercury, and they won't feed it to school kids. But I think adults are allowed to choose. And this isn't the only place where really high concentrations of mercury exist. This is a place in southwestern Norway. It's a little bit hard to see. It's a Google images. So there's like these fjords, these long inlets, and on one of them, there's very high concentrations of mercury in the sediments that come from extensive, discharge of mercury from a processing plant over a very long period of time. But interestingly enough, even though there's more mercury in the surface sediments here than there was in Japan, because of the difference in the diet, as well as the difference in the health status of the population, there was no Minamata Bay disease here. Right, it didn't occur, which also highlights something that we talked about last time, which is that when we look at whether we're looking at toxic effects that are lethal or sublethal, we have to think a lot about the prior stage of the population that is exposed. The population that is exposed is under stress for other reasons. For instance, post-World War II Japan, where there were nutritional issues and other stressors. Then that population becomes more sensitive to mercury poisoning than a population that isn't stressed at the same concentrations of mercury. There are other places around the world where we can find really high concentration of mercury. Mercury is used in gunpowder manufacture and explosives. So there's a notable site in New Jersey that's very contaminated. And of course, we have the mercury that we talked about previously from electricity release. This tends to be a lot less than these high concentration situations, but that's sort of how discrete high concentration of mercury can affect populations. So now I want to talk about tributyl tin. So remember, butyl is a 4-carbon alkyl group. Tributyl tin oxide is this compound. There's 3 of these butyls that are connected to a tin. It's also oxidized. And this is kind of what its chemical structure is. And the point about TBT, and some people also distinguish between TBT as the oxide or TBT as the fluoride, which has an F, is that purposely put into paint on boats so that stuff won't grow on the boat. When stuff grows on a boat, whether it's mussels or barnacles, then it makes the ship a lot less efficient in the water. It costs up more money in fuel and so forth. So this has been a pretty standard practice to paint things onto boats that would prevent what they call fouling. And so TBT was introduced in the 70s, and very quickly it was understood that this was a bad thing. It was having really negative impacts. And this is just a picture of a ship showing you some of the areas where fouling can have a really negative impact. You know, the propellers, the cooling pipes, the grating that brings water in and out, bow thrusters, which are things that allow ships to maneuver sideways, etc. And so, I mean, it's understandable that they would want to keep organisms off the ship, but the idea of putting TBT probably wasn't such a great one, because this is the contaminant that we were purposely introducing into the environment. It's organometallic. It has many of the same properties of mercury in its methylated forms, meaning volatile, fat soluble, bioaccumulates and biomagnifies. And it was being put into boat hulls so that it would leach out at a concentration and make sort of a toxic level of impact. It's still too early to know what the effects are on humans of TBT, but we do know there are lots of toxic impacts on other marine organisms, especially bivalves and filter feeders, as well as some seagrasses that live in and around harbors, where boats will spend a lot of time and therefore leaching a lot of this paint. And so this is a diagram showing you for this, bivalves as larvae and as adults, gastropods, copepods, crustaceans as larvae and adults, fish as larvae and adults, and algae. And then this is again a meta-analysis from a bunch of different studies. There's two different types of symbols. There's these open symbols and these solid symbols, and those are for different time periods of exposure. So what we see is that for really long exposure, greater than 96 hours, this is what the LC50s are, right? This is a log scale. So over a long period of time, a fairly low concentration can have an impact. If we're talking about an acute exposure, for instance, a spill or a much higher concentration event over a shorter period of time, then actually I'm saying that backwards. These are the ones that these are the yeah, I know I'm saying it right. Shorter exposure, higher concentration is required to cause the lethal effect. Longer exposure, you get the lethal effect with a lower concentration. But so you can see that there's a range. It depends not only on whether or not we're looking at larvae or adults, right? So look at the difference between that and that, or here larvae and adults, the difference between that and that. So the biological stage of the organism also matters. But for all these different organisms, these concentrations, these are measured in micrograms per liter. That's a pretty small concentration. That's a part per billion. That's pretty toxic. And so the sublethal toxic effects are many and varied, but a couple of them are, for instance, impacts on the growth rate of important provisioning organisms from a lot of coastal ecosystems, such as oysters. This shows you the weight of the oyster over some number of days of exposure with the concentration of TBT that they've been exposed to. So the control has a certain weight and it grew under certain conditions, and at increasing concentrations, you get less and less. At the same time, you get this over thickening of the shell, so that the weight of the organism goes down and the shell gets very thick. And so basically, the introduction of TBT wiped out lots of oyster fisheries and other shell fisheries just because of that. Now, there's an even more kind of infamous thing with TBT, which is something called imposex. So TBT is one of these endocrine disruptors, and it confuses the hormone systems of a lot of animals. Again, we don't know yet if there's impacts on humans, but this is an example of a whelk, which is a shellfish that, and the study was done in England. This is the concentration of tin that's found in the tissues in milligrams per kilogram. So again, those are ppms. And this says here, relative penis size. And so what this is the penis in females of the species. Because of the introduction of the TBT into the environment, the females grow male sex organisms, and then they're not able to reproduce. And so that has a long-term, in effect, is a toxic effect, because even though the organism doesn't die from it, it can't reproduce. So this is what we would call, sort of long-term effect. And this is a kind of a more recent report. This thing is called imposex. And so you can read all about it if you're interested. And this just, I put in, if you're interested, it sort of describes how this happens, the different stages of the growth. So, and TBT, because it is volatile and fat soluble, can be found in a lot of environments. So this is an example in southern England. So there's like sort of an inlet here, and there's a larger harbor here, and this is concentration of tin in seawater. Okay, and so here's a little legend. So that this kind of green hasher thing is the TBT. Green is organotin. So once this tin's been out in the environment, leached into the seawater, it can transform. The ethyl groups can be, or butyl groups, excuse me, can be broken down to ethyl and methyl groups, and some of it can be completely oxidized to total inorganic tin. And so you can see that in some cases, like right up here in this harbor, most of the tin is in this organic form. Some of these more distal locations, the tin, there's very high concentrations in the tin, but it's oxidized, meaning it's been transported there and then somehow transformed in the environment. This is perhaps the most telling example of how we know that these tin compounds are pervasive in the environment. This is a place in Japan. It's a palace moat in Tokyo that's not connected to the ocean, right? It has, there were never any boats in it that were directly leaching tin into it, and yet these are sediment cores going down. These are centimeters into the core, so 0 to 4, you know, something like a little less than 2 inches down, and then the next 2 inches, et cetera, et cetera. And so we can see that total tin concentrations are pretty high and pretty constant, and then these are different forms. So methyl tin chloride, Me is methyl, so it's a CH3 group, and then, you know, a dimethyl tin chloride, a trimethyl tin chloride, and an N-butyl tin chloride. In all of these cases, these concentrations of these various tin compounds are alarming because they shouldn't be there. They shouldn't be in that water because there were never any boats that were leaching tributyl tin into it. Instead, what this has to be is volatilization of tin from nearby Tokyo Harbor into the atmosphere, being partially transformed in the sediments to these other forms and accumulate. So it's not just that the tin, you know, problem, if you want to call it that, is localized to harbors. It can be local. It can be expanded to areas around. So that tributyl tin has been outlawed by treaty for most of the world. The U.S. Navy notably still uses tributyl tin. It's whatever, too valuable to them or whatever. But so after tin, you know, and mercury, copper is another one of these chemicals that has been introduced into the ocean in both kind of low levels and high levels, and is particularly harmful to marine organisms. They're not particularly toxic to us unless you get very high doses, but it is pretty toxic to a lot of marine organisms. And ironically, when the boat industry or shipping industry, whatever you call it, decided to ban TBT, they turned to copper as the thing that they wanted to put into paints to prevent the fouling. So copper is interesting because it's one of these elements here in the middle of the periodic chart that is solubility limited in seawater in both of its two common oxidation states, +1 and +2, unless there's organic matter present. And so this is, for instance, copper concentrations in the 1980s in the Gulf of Cadiz, which is Spain. And you can see the concentrations are sort of its color contour here. They hug the coast. And this is a common thing that is found, that the copper discharges, which were coming in here, they get caught up in the longshore current. They don't distribute offshore. They really have strong impacts on coastal ecosystems where there tends to be higher concentration of organic chelating ligands, which can help the concentrations disperse. And this, you know, kind of idea of copper-rich plumes has been found in a lot of different places. And so this is another part of England. This is southwestern England, Cornwall, basically, which is one of these places that ever since the Bronze Age, they've been like, you know, because they have the right rocks, they've been digging tin and copper and lead and zinc. various other metals that come along with them, cadmium, chromium, etc., and using them for whatever, making broadswords and so forth. And so there's fairly high contamination, variable but high contamination all around. And this is a study of specifically the effect of copper concentration in these estuaries. So distance from the mouth estuary, this would be at the ocean, and this would be 10, 20 kilometers up into the estuary, and the concentration of copper that is found in the organisms. And what we find is that depending on which one of these tributaries we're in, the concentrations don't increase by much, increase by more, or increase by very high concentration. And this is what happens after very long-term, thousands of years of exposure. There is a modification of the ecosystem where those members of the population that are able to handle the higher concentration, the hyposensitive ones, do better. And so basically the other organisms of that particular group die out. And so you can get these resistant groups. That doesn't mean that other organisms that might feed on that seagrass or interact with it in some other way don't have a negative impact. But over time, especially with the case of copper, which isn't as toxic, you can see those kinds of effects. This is just a picture of another kind of well-known, very high concentration copper spill, how it evolved over time. This is along the coast of the Netherlands, and so there was sort of a spill, and again, black is the highest concentration, and then lower and lower concentration. And you can see sort of the 31st of March, and then moving over into sort of, this one is the 12th of April, there's the 29th of April. It basically, this whole plume migrated along the coast. It didn't pull very far offshore, but the big fear was that it was going to get up into this area up here, which is the Wadden Sea, which we looked at once before when we were looking at heavy metals coming down the Rhine River. It's a big shell fishery, shallow embayed sea that, for a very long period of time, people have been taking shellfish from. And so, and the impacts of this copper still, even though the plume has, dispersed to some extent as it advected, it's still really high concentrations, much higher than you'd like to see. You'd like to see that be well less than 1. So the effects of copper on marine organisms are a little more complicated than things like tin, which we saw was an endocrine disruptor primarily, and mercury, which is primarily a neurotoxin. Copper can have all sorts of different things. It can greatly impact growth and development. It can inhibit photosynthesis, especially by impacting the electron transfer aspect of chlorophyll A. It can reduce chlorophyll content. It can increase cell wall permeability and affect the potassium ion pump. And so there's a lot of different ways in which copper can affect marine organisms, and it's not quite as acute as the other ones that we talked about, tin and mercury, but it's still something to be aware of. So the next element to talk about is cadmium. So cadmium is a really, really toxic metal. And, probably the most important positive use of cadmium was its discovery and use in pigments for paints in the sort of 1840s and 1850s. They were the first color fast yellows and reds and oranges. So sort of, if you're into art history, the whole Impressionist movement came about because of cadmium. Before that, painters were using organic dyes, which were not, or inorganic, like iron oxides, which just weren't nearly as bright. And so fast forward into around the turn of the 20th century, cadmium was being used in pigments for a lot of brightly colored items, and it was getting into landfills and trash and leaching into the environment. Cadmium is also significantly mined, not because they want cadmium, because they want zinc. And so cadmium co-occurs with zinc oftentimes in a lot of mineral deposits. So a lot of cadmium that gets released into the environment, and it's a very toxic metal. As far as we know, has very little to no health benefits for any organism. And it can affect a lot of different things. It can affect the respiratory system. It can affect the kidneys. It can affect the skeletal system. So cadmium 2+, which is the common form, it's a very similar ionic radius to calcium, so it can substitute for it. It has a different electronegativity, so it isn't, sort of a 1-to-1 substitution, but it does get into the skeletal system, and it can affect reproductive system in various ways too. So, and some of the details of this are listed over here, but it's considered a very broad spectrum toxin. So cadmium was another one of these things that was implicated in one of these very high concentration events that happened in Japan, and and this is it's similar but slightly different graphic. This is a river, and these dots are cases of something called itai disease, which is in Japanese, it means ouch, ouch. And so this was another one of these things that happened in post-World War II Japan, and this was a economically disadvantaged village, and so they were eating a lot of rice. And I'll show you in a second that cadmium was building up in concentration in the rice because, again, of a spill upriver. And then all these people were coming down with this disease, debilitating disease that was in effect decalcifying their bones. And but I think now, after the fact, people look at this and they say, cadmium maybe played a role, but it couldn't have been, it wasn't as clear cut as the Minamata Bay disease with mercury was. And here's the sort of the information about why that is. So first off, this is heavy metal concentrations in different tissues from one particular patient that had itai disease. And so remember we talked last time about differentiation, how different organs, different parts of an organism can accumulate different concentrations. And so you can see here, here's cadmium and co-occurring lead and zinc. These concentrations are astronomical in parts per million, right? 11,000 parts per million is a percent, right? Like 1.1%. So then you can see really high concentration in the ribs, so obviously bone, cartilage, other vertebra, and then lower but still significant concentrations in the viscera. And interestingly enough, also high concentrations of lead and zinc, but not always co-occurring in the same proportion, like lead and zinc, higher in relative proportion in the intestine. So then the other thing that was studied was the rice that people were eating. And so again, we've got the zinc, the cadmium, and the lead. And you can see here the sort of soil samples, the roots of the plants, and the part of the plant that people eat. And you can see that all three of these metals, the zinc, the cadmium, and the lead, are really concentrated in the root, which is not what part of the plant that people eat. But even still, there's very high concentrations in the rice itself that could have led to the exposures that caused these kinds of concentrations to occur. So the problem with this kind of association of cadmium with itai disease was that there have been other very high exposures to cadmium that haven't caused itai disease. One of them was a place, there was a study in southern Sweden where many people were studied from cadmium exposure, environmental exposure, and it was true that there was a decrease in the calcification of the bone density, osteoporosis, for people that had higher concentration of cadmium, that no one was experiencing this sort of itai disease. The concentrations weren't this high there. But there was a place in the 1970s where there was a very large spill of cadmium. This happened in the Derwent estuary of Tasmania. I think I've mentioned this before. It's Hobart, Tasmania is on a long estuary, and it's a really beautiful and fairly pristine place, but there was a metal processing plant sort of up from the town, and it's a fairly shallow river, and so for some reason a boat was going up there and it hit the bridge that goes across the estuary, and it leaked tons and tons of cadmium and zinc into the water. This is another one of these places where it's a shell fishery and a lot of people eat the fish there, and people were exposed to even higher concentrations than in Japan, and yet they didn't get the itai disease. So people now, they go back and they look at this and they say, well, maybe malnutrition was the primary cause of the Atai disease, and cadmium and the other heavy metals just kind of push them over the edge, something like that. Okay, so our next metal on our tour of fun metals is hexavalent chromium. This is our second to last one. After this, we'll talk about arsenic. So, chromium comes naturally, primarily as chromium +3. That's where you'd find it in the rocks here. It's pretty hard to oxidize to +6. You can, under certain circumstances on the surface of the earth, but it's not typically found in that form. But chromium-6 is used in a lot of different processes, kind of leather tanning and anti-corrosive coatings, textile dyes, wood preservatives, and we'll talk about the mixture of chromium, copper, and arsenic when we talk about arsenic. And so it's been known for quite some time to be toxic in its +6 form and not toxic in its +3 form. There was an example, for instance, of factory workers in the 60s that breathed in inhaled chromium-6, and after something like 40 years later, a quarter of the workers who worked at that plant had died of lung cancer. And various other studies suggest that ingesting it, meaning getting it in through your gastrointestinal system, can also be toxic. There was a sort of study done in China that was originally published in the 80s that showed toxicity at relatively low concentrations. And even here in the US, in the sort of 2000s, that copper has been shown to cause, chromium, I should say, when it's in the +6 form, has been shown to cause cancer. So I think we know that it's carcinogenic at certain concentrations, and what those concentrations are is still a little bit debatable, and so this is not monitored by the EPA, unfortunately. So, and you'll see in a second that Honolulu has the second highest chromium concentration in the country, but we don't know if it's +3 or +6 because there is no official requirement to assay between those two, and the standard is based on chromium +3, which is probably safe at concentrations of 1 million times higher. So there's been progress at studying chromium, but not as much as there should be. So this is a map. This is from the USGS, Geological Survey, showing you chromium concentration in water. This is chromium-6, so specific that specific form of chromium, which is especially toxic. And you see kind of a interesting. There's some states that obviously don't report. That's why they're in white. But you'll see some areas, especially in the arid Southwest, where the chromium is higher. This tends to be a characteristic of the sort of deeper aquifers where the chromium is found, and deeper, coarser aquifers is part of why we see the concentrations. So when we talk about arsenic, we'll see similar pattern with that. The highest concentration in the country is here in Oklahoma, northern Oklahoma, but the second highest, as is shown on this map, is Honolulu. And this is another plot sort of showing you, this is from the California EPA, showing you concentrations, and here we are, second on the list, Honolulu. And interestingly enough, the kinds of things that you expect from copper, especially ingestion through water, don't occur here. I mean, we don't have cancer clusters with stomach cancers and whatever, so it's obviously a little bit more complicated, but probably we're getting way more. If you're not filtering your water at all, we're getting more chromium than one would like. Now, chromium-6 was made famous or infamous by the Aaron Brockovich movie. Maybe you've seen that, maybe you haven't. Julia Roberts played this kind of reluctant environmental activist. Basically there was Pacific Gas and Electric, that's what PG&E stands for, which is the kind of main electricity provider in Northern California, had some subterranean spill of chromium-6. They were using the chromium-6 somehow in cooling water in a electricity generation facility that was in a kind of a rural town, and it spilled into the water, and there was like 60 years of contamination of the drinking water. The company was like, I'm sorry, but we don't know what's going on. It's not, people were getting sick and they were claiming it wasn't that, and it was lawsuits and all this kind of stuff. And it was, basically decided that they have to clean it up. And the city is, town is basically gone now. No one wants to live there anymore. But it's one of these things where because it's kind of on the cusp of the clear-cut evidence, and it's not so clear-cut evidence. And so even though the EPA says, yes, you need to clean this site up or whatever, industry scientists have thus far been able to sort of argue against full cleanup and full toxicity. And so it's one of these kind of interesting stories if you want to look it up and read a little bit more about it. But we do know chromium-6 contamination at high enough concentrations where it's, for instance, visible, like this greenish water. This is in the Himalayas. Again, similar kind of aquifer settings to what we have in the Southwest, and very high concentrations of chromium have dermatological issues, abnormal hematological function, as well as some of these gastrointestinal issues. So we know that at some concentration it's definitely toxic, but the question is at what concentration and at what level of exposure. And so it's been kind of mired in the multiple decades of inaction by the EPA. But I personally would filter my water. Well, I do filter my water, but I would filter your water. It doesn't take much. Just even I don't want to promote a particular brand, but the ones that have the activated charcoal, they pour the water in, and those kinds of things are fine. They will take out most of that chromium. So last thing I want to talk about is arsenic. So arsenic is another one of these elements. I think people recognize arsenic is a contaminant. You find it in rat poison. It can kill organisms. It's one of these ones that you hear about, people, spouses poisoning each other, and it builds up, and you can find it in your hair and in your nails if you've been exposed to it. But the sources of arsenic into the environment are multiple and varied. They've changed over time. It's become much less common to find arsenic contamination, but here, for instance, in Hawaii, it's all over the place because it was used in the past. So before the 1960s, it was very common in pesticides, especially for pineapple and sugar. It's still used, but in a much more limited way. But arsenic is also very important in semiconductor manufacture, and much like mercury, when you burn coal, there's a fair amount of natural arsenic in there, and so it gets released from the coal during burning and accumulates in the environment. There are also, arsenic-treated wood products, and we'll talk about what this is, but it was basically a treatment that would make wood sort of not dry rot. And this went into a lot of houses. If you have a house, like I have a house here that was built in the 30s, it's full of arsenic-treated wood. That's just sort of how it is. And finally, there are some natural, kind of sources of arsenic that are pretty high concentration, one of which is found in deep groundwater, which we'll talk about as we get near the end of this particular topic, that can introduce very high concentrations for non-contamination. So the first sort of way that we come in contact with arsenic is this CCA wood, chromated copper arsenate. Basically, when you go to the store and you see treated wood, up until, it's not so much anymore, it's been about 20 years, but that was that green colored wood. It was being injected under high pressure and used for everything, the framing of the homes, the jungle gyms, kid stuff, fences, especially anything that came in contact with soil. And this became really popular in the 70s. But by about the turn of the century, it was outlawed for many uses. It could still be used as part of the internal part of the structure, like the framing that people don't come in contact with. But just imagine you're a carpenter and you're sawing this wood all day and you're making dust, and that dust is distributing in the environment. It can have a pretty negative impact. You can get a very high dose of copper. There are percents of arsenic in these materials, and so now it can be used in certain cases, but it can't be used for, like, children's play structures or schools. It can't be used on picnic tables, residential fencing, et cetera, et cetera. So this exposure pathway has been limited. And just a replacement, most of the treated wood is with boric acid now, which is a lot safer, not without its problems, but a lot safer. So now I want to talk a little bit about arsenic in groundwater, which has been another really serious contamination issue. And for this, we have to recognize some of the chemistry of arsenic. So this is a PE-pH diagram, pH across here, PE over here, EH over here. And so you see, I know it's been a while since we talked about these in the class, but hopefully you can see sort of two things to color coded it. There's an oxidized form of arsenic, arsenate, and there's a reduced form of arsenic, arsenite. And the transition between them happens about midway through in this field, right? When we get to high pH, it happens lower. And at low pH, it's a little bit higher up. But if you remember at all anything about some of the other elements we've talked about, this is around the same place as the iron 3 to iron 2 transition. The sulfate to sulfide transition happens more like down here. So this reduction will happen. It's similar redox potential to what iron reduction is. Then we also have these things, if you recall, vertical lines on EH-pH diagrams are acid-base reactions where there's no electron transfer. And so arsenous and arsenic acid are directly analogous to phosphoric acid, H3PO4, triprotic acid, and we've got H3AsO4, right? So it can lose one hydrogen ion, become this singly charged compound ion, doubly charged, triply charged, etc. And in this form, the form that's shown here in red, is very similar to phosphorus. So all the stuff we talked about, phosphorus binding to iron, binding to aluminum, arsenic does the same thing. When it reduces, it takes a different form. Phosphorus doesn't have this analogy because it doesn't have the two different oxidation states, but it also has a pH dependency to its forms. But you can see here that whereas we make the anion at relatively low pH, we don't get into anionic forms until we're at high pH for the reduced arsenic 3+. So this is a plot showing you the binding of arsenic to iron in this is laboratory studies, but they're meant to mimic either soil particles or particles of substrate in the groundwater. So you can see here pH and the percentage of arsenic that is absorbed to surfaces. So the first thing you see is that arsenate, the one that's the oxidized form, that's like phosphorus, binds 100% all the way up till about pH 8. And then it starts to unbind. And the reason it's unbinding is that various iron oxyhydroxides reach their 0 point of charge and then flip their sign when we get to very high pH. So they go from being positively charged, which a negative anion will bind to being negatively charged themselves, in which case the arsenate anions don't bind anymore. Now, the arsenite, the reduced form, it doesn't bind as effectively, right? And part of that has to do with the difference here in the pH, right? We have a lot of neutral arsenic. We only start to get some negative charges when we get higher. So it's less effective at binding. It can bind to slightly higher pH, but the key thing is that when pHs get above 8 or 9 or so, we pretty much don't have any binding left. And as you can imagine, if we go from oxidized form to reduced form, even at more modest pHs, there's going to be a release of arsenic from being bound to not bound, at least for a fraction of it. And so these are sort of a reminder of a slide I showed you earlier when we were talking about phosphorus. And what I want you to do is just think about arsenic in its oxidized form can take the place of phosphorus every place that you see a phosphate anion, you could have an arsenate anion. So it can be bound to iron, either, you know, monodentate or bidentate. If you recall, this form of phosphorus was kind of reversible, whereas this form of phosphorus binding is irreversible. The same thing for arsenic. The only way you can get this to unbind is to reduce that iron from +3 to +2, where it becomes solubilized and then it will release. Arsenic can bind to organic ligands. Arsenic can, as is shown here, again, as sort of a monodentate or bidentate complex. It can be bound to iron, it is bound to organic materials. It can be bound to aluminum, it is bound to organic materials. It can even be sequestered into inorganic minerals, although this isn't as common as it is for phosphorus. But basically, the arsenic cycle and the phosphorus cycle are very, very similar. Okay, so another interesting thing about arsenic is that because of its great similarity to phosphorus, there have been, and there's a fair amount of arsenic around in the environment, there have been mechanisms, biological mechanisms developed over time that allow a lot of organisms to detoxify to a certain point, right? They can deal with a certain amount of arsenic and it's not, you know, too much of a problem. There are even some microorganisms, especially archaea, but some species of bacteria as well, that basically reduce arsenic. So they take arsenate and make it into arsenite in the same way that other organisms take sulfate and reduce it into sulfide. And you know, these are called dissimilatory arsenate reducing prokaryotes. They don't occur in all environments, but they do occur in environments where arsenic is relatively high in concentration. And one place we think that these operate is in deep groundwater aquifers. So they take the oxidized form and they make it into the reduced form, generating energy for their metabolism. And the most important aspect of this for our purposes is that arsenic goes from being this oxidized, highly bound form to a form of arsenic that is less bound into surfaces and therefore can be available in the water for potential contamination of drinking. And this is sort of how the, this is, you know, a well, and this is sort of how the combination of arsenic reduction and iron reduction kind of work together so that because they have a roughly the same PE of transformation, whether we have organisms that can take arsenic 5 and turn it into arsenic 3 or not, we almost always have organisms that can take iron 3 and make it into iron 2. And so some combination of arsenic being reduced from arsenate to arsenite, as well as iron reduction, causes arsenic to go from being bound on a substrate to being free in the water. So this is a map of arsenic concentrations in the US. And as I said before, it's a USGS map. It's sort of, to a very first order, similar to the chromium, in that we have a lot of higher concentrations, these sort of warm colors in the Southwest. There's also some high concentrations up here. It has to do with the type of sediment that is present, the sort of glacial lust deposits. Some in Alaska, Hawaii does not have, we don't have much arsenic in our rocks naturally. And so, you know, it's a complicated map, and there's more than one reason to have relatively high arsenic concentration. But interestingly, this is another one of these elements that there's been a lot of politics involved with. So when at the very end of the Clinton administration in the late 90s, they decided to lower the threshold from something like 50 ppm down to 10 ppm, and which I think was justified based on the science. As soon as the Bush administration came in, this is like before 9/11 or any of that stuff, like one of the very first things was to just sort of arbitrarily say, nah, we don't like it. We're switching it back up to 25. And the reason was that there were a lot of municipalities that said, oh, arsenic is high in our water naturally, and it's going to cost a lot of money to clean up, so we don't want to do it. The thing is that whether the arsenic is there naturally or not naturally, it's still a contaminant and it's still hazardous for people. You can argue about whether or not the money that would have been spent to meet the 10 ppm standard versus the 25 ppm standard could have been better spent on a different contaminant, but that wasn't the motivation for that change. And so that we currently have a 25 ppm standard, which is not low enough. So for places in the world that have it, we know it's a problem. And this is the sort of the classic example of what really brought arsenic to people's minds. So in the kind of 70s, 80s, and into the 90s, the British, who had, as part of their colonial past, Bangladesh and India were part of their empire. They had gone in with the good intention of providing fresh, deep groundwater to populations because the surface water was very contaminated. They pulled up a bunch of water, and very soon thereafter, people were starting to have all sorts of signs that were pretty clear were from arsenic toxicity. And what it turned out to be is that they were sampling very deep water that were below this iron-free, iron-2 oxidation front, and therefore had arsenite instead of arsenate. And so while the water may have been cleaned from the perspective of biological contamination and other shallow occurring contaminants, they were introducing very high arsenic concentration. So throughout the sort of 90s, especially the last half of the 90s, there was a lot of effort put in to try to figure out why is there arsenic in the water? And it was basically determined that there was an association between arsenic and iron. There's also at high enough concentrations association of arsenic and bicarbonate ions. And this is a map of plot, I should say, of some well logs of sort of showing you the different lithologies. So like sand and clay, remember this would be fairly impermeable, this would be fairly permeable, water's flowing, and a whole bunch of different parameters. And the key thing is the dissolved arsenic concentration. Dissolved arsenic concentration coincides with all of the redox ladder transformations that we've been talking about, the presence of methane, the presence of ammonium, the presence of iron, above, shallower than the place where sulfate reduction starts happening, where the concentration of sulfate is occurring. And so, I mean, this is something, and we'll talk about this next time, we're talking about water treatment. This arsenic can be removed from the water. It's not like a fatal flaw for this effort to provide deep groundwater, but it's something that they didn't think about at the time that now people do think about, and this water can be treated with iron and be oxidized, and then the arsenic can be pulled out before it's distributed to people is one of the many steps. And we'll talk about those steps next time. So that's where I wanted to leave it. And are there any questions about this? Probably I'll give you the midterm on Thursday, but it may not be until Tuesday. Thank you very much.